What is a polymer? We can break down the word into its two parts, with poly meaning many and mer meaning units. From this we can understand that a polymer is a molecule made up of many units bonded together. Polymers can be either synthetic or natural. Some examples of synthetic polymers include the polyester in your clothes, the polystyrene in foam cups, and the PVC in plastic pipes. Natural polymers include DNA nucleotides, proteins, and cellulose. Polymers are everywhere, appearing in both everyday life and cutting-edge technology. Here are some more applications and examples of both natural and synthetic polymers. Almost everything in the human body and in nature is made up of polymers. Some examples in the body include your muscles and skin. Other examples include rubber and cotton. On the synthetic side, polymers are used to make a wide range of things. Plastic beverage bottles and fishing line are some things you may have used that are polymer-based. Other common items made with polymers are rechargeable batteries and electronic screens. Polymers are also very important in high-tech fields, such as aerospace. While polymers are all based on a general structure of linked monomers, they can have very different architectures and compositions. Let's start with composition. The simplest polymer composition is the standard homopolymer, which contains only one type of monomer unit. When another type of monomer is added, you form a copolymer. In copolymers, the monomers can be arranged in several ways: randomly, alternating, or in blocks. Now let's talk about architecture. The monomers can form a linear chain, or they can have more complex architectures involving branches or rings. So which polymers do we study? Our research mainly revolves around block copolymers. These are polymers comprised of two or more homopolymer blocks bonded together to form one larger polymer. By tuning the composition and architecture of these polymers, a variety of molecular structures can be achieved, as shown here. And these various types of polymers can further assemble into larger supramolecular nanostructures. Why are these supramolecular nanostructures important? One of the most important parts of materials science is the relationship between the structures and properties of materials. This is the materials tetrahedron. It illustrates how the four components of material science are all related to each other. For example, the performance of the material is dependent on its properties, while the properties themselves are dependent on the material's microstructure. By understanding this relationship, we can optimize a material's properties and performance by designing a specific microstructure. We can even combine multiple structures within a material to combine or enhance the overall properties. So how can we achieve the designed nanostructures? Nanostructures can be formed via two main methods: top-down and bottom-up. Top-down approaches involve selectively removing material from the bulk mechanically or by exposure to light, electron beams, or X-rays. You can think of it like a sculptor chipping pieces off a block of stone to form a statue. Bottom-up approaches involve spontaneous or directed assembly of molecules into larger structures, like building with Legos. Each method has its limitations, and we can choose different methods based on the material we're working with and the structure we want to form. For example, it can be difficult to create structural features smaller than 20 nanometers with a top-down approach. Bottom-up methods may be better able to create these smaller structures. Our lab primarily uses a bottom-up approach called self-assembly. What is self-assembly? Let's start with one block copolymer. Let's call the two blocks A and B. For many block copolymers, the two blocks tend to separate from one another. A prefers to interact with A, and B prefers to interact with B. So when you take a bunch of these polymers and put them together, they will move to separate into larger regions of A and B. When forming these regions, there are two forces at work. As we discussed, the A and B blocks have unfavorable interactions and try to separate from each other as much as possible. But at the same time, the movement of the polymer chains is restricted because 1, the two blocks are covalently linked, and 2, the chains prefer not to be stretched too far or compressed too much. Therefore, a balance is formed between these two restrictions. Several other factors like the volume fraction, composition, architecture, and temperature can affect this balance and cause a different structure to form. Here are some common structures formed by diblock copolymers. Lamellae form when the amount of A block and B block are relatively even. As the volume fraction of one block is increased, we can start to see structures like the double gyroid, cylinders, or spheres. Our lab primarily works with the double gyroid structure. Things get even crazier when you add a third block. Here are a few examples of some cool structures that form from triblock copolymers. Now let's go back to the double gyroid, our work's primary focus. This structure is an example of a tubular network structure. These structures consist of two intertwined networks of one polymer inside a matrix of a second polymer, shown here as the empty space between the networks. The two most common tubular network structures are the double gyroid and the double diamond structure. So why do we care about these network structures? These structures have several useful features. For starters, all the individual networks are 3D continuous, meaning you can start anywhere within a network and get anywhere else within that network without crossing any boundaries into another domain. The matrix surrounding the networks is also continuous. These networks also have a very high surface area to volume ratio. This leads to all sorts of useful properties like mechanical stiffness, transport of gas or charges, and unique wave interaction properties. We can even remove one of the domains and fill it with a new material such as a metal or ceramic. A specific application of the double gyroid structure is a new type of battery. Using nanotechnology, a double gyroid network can be created as a battery with one network as the cathode and the other as the anode. These double gyroid batteries are many times more efficient than a conventional battery that uses cathode-anode layers. High-efficiency solar cells can also be made using double gyroid structures. Something else we look for in these structures are defects. All the images we've looked at so far show perfect structures. In the real world, however, we can find lots of different defects in our materials. Defects are important because they can significantly change a material's properties, which is why our lab is interested in them. There are three main types of defects. The first are zero-dimensional defects. Also known as point defects, these defects affect an isolated point within the structure. However, in self-assembled polymers like our double gyroid networks, these defects instead affect larger areas than a single point, such as a single junction within the network. Next, we have one-dimensional or line defects. These defects affect the material along a line and can sometimes be curved, forming a loop within the structures of the material. Finally, there are two-dimensional or surface defects. These defects include external surfaces and grain boundaries, a type of internal surface between two regions with different orientations. In this grid, these are our two grains, and this is the grain boundary between them. In double gyroids, however, the usual 2D grain boundary defects look more like a volume with a short-range ordered networks. So how do we study these structures? How do we even see them when they exist on the nanoscale? To view and study these materials and structures, we need high-powered microscopes and instruments. Traditional methods like small angle X-ray scattering and transmission electron microscopy can get us structural data and 2D images of the material. However, to study these complex 3D networks, we need a good way to analyze and visualize the structure in 3D. For this, we use a method called slice and view scanning electron microscopy. With this method, we can get a 2D image of the surface of a sample, then mill away an extremely thin slice of material we're studying, and get an image of the surface after each slice. Then we can put the images together to reconstruct the 3D structure of our material for clearer in-depth analysis. Here is an example of a slice and view reconstruction of a double gyroid network. The advantages of slice and view are that it can provide large volumes of data compared to normal data collection methods, and it gives us high-quality, detailed reconstruction of the data, especially when compared to simulation data. To see more examples of the cool stuff we study, go to our group's YouTube channel at Thomas Group. Block copolymers are cool.